DNA replication. Now, in DNA replication, your double helix structure will be there inside your body, right inside your cell. And then what will happen is that this double helix structure will unwind. So from being a helix structure like this, it will unwind and become two strands next to each other, right? So the first thing that is going to happen is that your double helix structure will unwind. So we are talking about DNA replication now. How do we get our sequence inside of ourselves? Now, in DNA replication, the double helix structure will begin by unwinding, meaning it was helix structure like this, two strands in a helix form. They unwind, and then you just have two strands that are next to each other. Now that you have these strands next to each other, remember they are connected by hydrogen bonds. What will happen is that these weak hydrogen bonds will now break, right? They will be broken by some sort of enzyme. The hydrogen bonds here will now break, so that the adenine is no longer attached to the thymine, so that the adenine is no longer attached to the uracil. So they will start, if they are not attached to each other by these hydrogen bonds, because these weak hydrogen bonds are starting to break, they are going to unzip, because they were locked together just like a zip by weak hydrogen bonds. Now once these hydrogen bonds are broken and they are not attached to each other, they will start to unzip and go their different ways. But the interesting thing when they go their different ways is that they still carry the genetic code. They still carry the sequence. You remember that the T was attached to the A. The thymine was attached to the adenine was attached to the thymine, the guanine was attached to the cytosine, the adenine was attached to the thymine. So this strand will basically remember what this other strand sequence was, because it still carries this sequence of nitrogen bases. Now it still has A, T, C, T. That strand that went away will still have those nitrogen bases that would have bonded with them, and still have T for the A for the T, C for the... So they basically remember what the other strand sequence was. Now, we now have this single strands that are going their separate ways. We call them templates, right? They are basically now a template that remembers what the other strand that was attached to it, how it was sequenced. So this now is a template. Number 3, a template, and now this is a template. What it is going to do now is that this template will now be looking for this strand, but this strand is no longer available for it. So it will now search in a nucleotide pool. This, we call them nucleotides. So we call this the nucleotides. Now, this template, the red one, it's going to go in the nucleotide pool. Nucleotide pool. Is there an E there? I don't think so. I'm not sure. It doesn't matter. You go check the spelling. So it will go into a nucleotide pool whereby these nucleotides, the guanines, the adenine, are just swimming in there, but they are not attached to anything, right? Now, they will come and form a strand here. And then they will sequence themselves in a way that they can attach to this template. So this template used to be G attached to T. But now that this T is no longer available, this new strand is gonna come and then provide a T from the nucleotide pool so that it bonds with this A, right? And now the same thing happens to the T. Because this A is no longer available, this new strand is going to come and provide the A from the nucleotide pool so that it can bond with the T. And now we call this the complementary strand. This strand is called the complementary strand because now it comes with nucleotides from the nucleotide pool and it sequences them so that they can attach to this to our template here. So this C that are attached to the G is no longer available. So this the complementary strand will provide the C. This G is no longer available, so it will provide a G. Same thing with this side. Now, because this A is no longer available and will be attached to a new thymine, this new strand needs to provide an A for the T. The A, its T is no longer away. This is no longer there. It went away with that strand, so it needs to provide a T. This one needs to provide a G. This one needs to provide a C. Now, what started as one DNA, I don't know if I'm allowed to say that. Let's just say what started as one DNA will now be two identical DNAs. So now we will have this DNA AT, AT, TA, TA, GC, GC, CG, CG. They are now identical to each other, and now we have two of them. It's called DNA replication. That's how we make more. So now that we have this one, it will then go back and become helix again, and now we have two different strands. We say we have two identical DNA molecules. And that's our 5. So after they copy themselves like that, we now have two identical. You can see that I put effort here. TA, TA, GC, GC, CG, 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 CG. So there are now two strands, two molecules that are identical to each other. Now, if I have a baby or anything, then I will provide them with this one and keep the other one, and then it will have an identical DNA, but there will be some mutation or whatever, and then they will have their own distinct sequence because these are so small. The strand, one person's DNA, if you stretch it out and put it in one line, it could probably go around the world like 4 times. I'm not sure how much, but I know it's a lot. So that's how our DNA replication happens. Our double helix structure unwinds, becomes just two strands next to each other. Our hydrogen, the weak hydrogen bonds that are linking up these two, that are linking up the strands, remember now they are back and connecting these two. But here they will be broken, and then once they are broken and the two strands are not attached to each other, our DNA will unzip, and then we'll have two template strands, and then these templates will search in the nucleotide pool so that it gets a new complementary strand that makes an identical pairing, just like in here and in here, and now you have two identical DNA molecules. Now, what's the point of this DNA replication? Remember I said we need to produce more. It doubles the genetic material so that it can be shared between the resulting daughter cells during cell division. During cell division, not just division. During cell division. So when the cells are dividing, the new cells will now share the genetic material. So since you had one cell and now they divide to form two identical cells, now this genetic material that was in here, it doubles by DNA replication, the way I just explained, and then one will go in here, one will go in here, and now you have two cells. The same thing will happen, and then those will do that again, and then it will be an endless process. Also, results in the formation of identical cells during mitosis. The same thing, almost the same thing I was explaining there. And that's DNA replication, the structure of DNA and RNA. Remember that with RNA is just one strand like this instead of a double helix structure. We will compare them and discuss more in future videos.